Today we have Dr. Andreas Westphal. He's an associate professor of Cooperative Extension in Nematology at UC Riverside. And today he's going to be speaking on nematodes, managing nematodes in nut crops. So now I'm going to go ahead and pass this over to him. So you can go ahead and share your slides.
Well, good afternoon, everybody. Thank you very much, Cheryl, for this introduction, and Peter for having me on this. So it's with great pleasure that I want to share some information with you on managing nematodes in nut crops. And when I show you examples today about that, I will give you examples from almond and walnut, because although nematode pressures are a little bit different between the two crops, yet there are similarities that are worth exploring in a talk like this. So when we talk about nematodes, and of course we need to know why are they even of so big concern to us, because they are living in a soil environment where we can't easily detect them. So oftentimes we only see the symptoms or the results of the feeding. And we need to consider how to monitor for them, because they are out of a regular frame of reference, what we explore. Now, then we want to talk about some rootstocks, because yes, it will be the most efficient way if the material, the plants that we put into the soil don't even get hurt by nematodes. But unfortunately, many of the rootstocks that we currently can use are not that fit, and we need to use pre-plant soil treatments to establish a vigorous root orchard. Now, many folks, of course, are also interested in post-plant mitigation options, because of course, once an orchard is in the ground, a lot of investment has been made, and it's important to then try to maintain high productivity in such an orchard. And although we are in the middle of a drought, cover crops are still a very important topic. Many of you have heard about it under the soil health initiatives, but I also wanted to contribute some information on plant-parasitic nematodes and what the role they may play there. When we think about nematodes as agronomists or production folks, we mostly think, of course, as of plant-parasitic nematodes. But just to give you an idea, not all nematodes are really detrimental to us. Most of them are marine, so pretty much play no role for us in this particular context in plant production. But there are also about 25% of the species known that we call free living. And those are nematodes that are either feeding on bacteria or they're feeding on fungi. And they are very important for the soil health and also for the nutrient turnover in the soil environment. Only 10% are very damaging, or can be quite damaging. And we want to see what the different feeding habits of these guys are, since they are living underground. And the ones that we are foremost interested in California are root-infecting nematodes. And if you go from the left to the right on the schematic, on the top row, you see Melodogyne, or root knot nematode. That's a nematode that is only mobile in its infective stages, but once it enters the roots, it becomes what we call sedentary. It just doesn't move in the root tissue anymore, and it stays at that site where the nutrients are transported to the nematode itself. So the nematode kind of tricks the plant into creating what a physiologist calls a new sink, where the nutrients are being delivered to it. Now, it's a very intricate interaction of the nematode and the plant to accomplish something like that, because cells need to be changed within the root tissue, and the nematode needs to do that with its enzyme stylet that and the corset that it uses for that. Now, because it's such an intricate system, it's comparably easy, quotation mark, to develop resistance to such an infection, because it's such an intricate cascade of events that if one step is interrupted in there, you may have already resistance. So it's comparably easy. I don't call it really easy, but compared to others. Because if we go to the center root, we see Platylenchus, and you have heard about Platylenchus vulnus, or the root lesion nematode. This one is infecting. It can, although we call it an endoparasite, a good portion of its population is in the soil, the other portion is in the root, and this nematode continues to meander within the root tissue. So it never has to really fully change root tissue, it just feeds on cells and then moves on and finds others. So the interaction is a little bit more casual, I'd call it, compared to root knot nematode, and finding resistance is a lot more challenging, because the target that the plant has to find to not allow the nematode to do is more difficult to find. Ring nematode on the upper right of the schematic live their entire life within the soil and only feed from the outside on the root tissue. So here the interaction becomes even more careful. And yes, it's very difficult to find resistance to ring nematodes because there's not this intricacy of those two things. And if we look, for example, in prunus for rootstocks, it seems to be reflected what I just outlined here, how rootstocks, there are plenty available who can deal with root knot nematode reasonably well. There are much fewer in root nematodes. I would even state sustainably resistant for many years is not really available in current rootstocks that are out there. And in ring nematodes, people who have that problem are of course aware that Lovell would be an issue there, or would be a tool there that shows some tolerance to it, but high levels of resistance and high level of tolerance are difficult to find. And mind you, Lovell would be extremely susceptible to root knot nematodes. So in that thing, this ring nematode is very hard to deal with. The next thing that is so critical in perennial crops is that many times nematodes occur in different assemblies. So here you see a few nematodes, the dagger nematode, ring nematode, the cigar-shaped one with all these structures on the outside of its body, and then root knot and root lesion nematode. So many times these nematodes co-occur, and that makes it so difficult because one has to address several nematode problems at the same time. The next problem is that these nematodes in tree crops are at least 5 feet deep in soil. As you see on this schematic, you see that at 0 to 1 foot depth, you have even lower population than if you were to drill deeper in the soil, close to 3 to 4 feet, where population densities of one nematode, in this particular case, pin nematode, can be much higher than even at the surface layer. That per se really impacts how we deal with nematode problems, because of course a tree root will grow to those depth layers and then will be impacted by nematode infection. At the same token, it becomes very important when we renew an orchard and go from one to the next, when we time our sampling. And since today is about how to monitor for nematodes, at the time of orchard removal and renewing the orchard to plant a new one, there's one very effective time to reduce nematode numbers. Now, the time for sampling should be best done in the old orchard when it's still in place, because then we can sample at sites where we know that there are young tree roots and get our nematode or soil sample for nematode detection that way. The least favorable time would be August or September, and not only because of the thrilling hot weather conditions that makes the sampling so exhausting, no, also because the nematode in the upper foot or so, where the soil pretty quickly dries up in a fallow field, can be dried out so quickly that the nematodes succumb, although there are then still population densities deep in soil. For this example, at 3 to 4 feet, soils dry out much slower, may still have sufficient moisture, there may still be live fruits, so nematodes will still be there. So if you don't get your sampling done at the right time, you can still do it later in the year, but then you will be forced to sample deeper in soil to find remnant population densities. It also impacts how we do our soil application of pre-plant options, because if we know that the greatest population densities are below 3 feet, let's say, well, then we need to also reach those sites with our treatments. That's why there are prescriptions for doing soil fumigation in dried out soil, as Mike McHenry has shown that that's necessary at certain moisture levels. And that's why we do a lot of our soil treatments with water drenches, but we're going to get to that in just a minute, to deliver the material to the site of action. So it's very critical to understand these bases of nematode behavior in the environment. Now, we can't go into too much depth about nematode sampling today, so that's why I put up here a YouTube link where you can learn more about that. A couple of years ago, we made a video on what to all consider when you're working on soil sampling for nematode detection. Now, when we select our rootstocks, be it in mostly in pruners where we have more choices, but also the same thing remains true for walnut and other tree crops, of course. We have many parameters that we need to consider. I mean, if we are in a windy area, we need, of course, good anchoring of the trees. Then vigorous rootstocks are frequently liked, at least in the current production systems. Then, of course, it's about nutrient uptake or the aversion of boron or salt problems. But it's also a question of availability. So what does a nursery have available of the scion that I want is growing on? So this can all be improved to a way when we plan more. If we plan at the time that the trees are produced, we can get the rootstock mostly that we want and need for our particular field. But of course, if we buy the tree shortly before we want to plant, then we have other challenges there. So resistance to soil-borne pathogens, although in the view of a plant pathologist like myself, very important, should perhaps receive a little bit more attention than it has in some instances. So where this went totally haywire is an example here, where people used, of course, their best deciding factors to find out what rootstock to use. And this is up in the Chico area where it's very windy. So folks want something that is anchoring well. Crimson 86 is known to anchor very well, has very strong roots for that, but it's also known to be very susceptible to nematodes. And as you can see here, you see the typical symptom of nematode damage, that you get uneven growth of the trees. And if you look at this poor fellow here, which really doesn't want to put on any growth anymore, yeah, you're concerned, and of course the orchard is in jeopardy for something like that. Now, when we backhoe those out, we saw all these huge galls on there, which are, well, dove egg-sized galls on there, and this root system is no longer functional, and that's of course why the tree wasn't growing right. Whereas when it's a healthy tree, you have these nice anchoring roots, of course, and you have a number of fibrous roots on there as well. Now, how do we get to better rootstocks? It's a long-going process, and I mean, many times people say, well, why does it take you so long to have a better rootstock? Well, because it's a long-going process. Here's an example. In walnuts, we plant saplings, as you see on the upper left-hand corner. Then we grow them for 1 year after we had inoculated them 1 month after planting, and grow them for a 2nd year. We found early on that doing any kind of fruit sampling after 1 year of growth is well, more or less of a wasted effort, because numbers can be quite deceiving, and we do need 2 years of growth under field conditions. So that alone is already 2 years. And from there, though, we were successful with the breeding orchard that I inherited from my predecessor to make selections where you here on the left see a tree that is a current commercial standard of walnut rootstocks in the nematode infested and replant problem site. And on the right, you see a site where my assistant is standing right next to a selection of ours. So this is just rootstock material. So it doesn't automatically convert into a bigger commercial tree, but of course it looks very promising that a rootstock can handle these conditions so much better. And so we took genotypes from there. Trees and walnuts are pretty much exclusively in our research program now produced as clonal plants, meaning selected crosses are being made. The seeds from there are via embryo rescue and clonal propagation made into new trees. And then we can test them under field conditions. But the beautiful thing is that every one of our genotypes still grows in a tissue culture lab in Davis of the walnut breeder, who then can make new plants as the need arises. We took advantage of that by taking front runners like the one I just showed you a picture of, planted into non-fumigated and fumigated ground here at Kearney after they had been made into full bare root trees. And you see in the upper left-hand corner, the first year after planting, they were all planted in 2018, where there was hardly any separation of the different genotypes, how they grew under non-fumigated or fumigated conditions. In the 2nd year, those differences became larger, as you can see, so they stretched out more. And in the third year, that's all the data that I show on this today, the non-fumigated all were much lower than the fumigated one in rootstock diameter and scion diameter, but there was a separation beginning between those within the trees that were growing in non-fumigated ground. And so these two here, for example, were much better than others, even under non-fumigated conditions. And although they didn't quite reach, at least from an overall tree size and growth, the potential in fumigated conditions, they did grow much better, which was then shown in this slide here, which is, well, a little bit busy, I guess, but it's a nut weight per tree. And it's shown for trees in fumigated or non-fumigated conditions, the blue being the non-fumigated conditions, and the red being the indication for each of those rootstock genotypes under fumigated conditions. And as you go through there, you see that pretty much all of them yielded better when they grew in fumigated ground. Not a big surprise there. But what was impressive to me, at least, was that the experimental 6 that you see had much higher yield under fumigated conditions than the commercial standards currently do. So certainly something to pursue if we continue having soil fumigation. What was more important, more the purpose of this whole study, was that the experimental 6 also had yields in non-fumigated ground, which were at least comparable to those that were reached by other, the current clonal standards, under fumigated conditions. So it has a much higher yield potential, and because of its constitution of resistance and tolerance, can much better produce yield, and that way can even be competitive under non-fumigated conditions, which may or may not be the future for us at some point when we don't have fumigants anymore. So out from this, we continued our process on walnut rootstock selection, and we had 2 big breeding populations where the breeder Pat Brown and his predecessors and Chuck Leslie produced breeding populations of 2 mother trees that were crossed with particular pollen. From there, they went through this clonal propagation process I just described, and then Greg Brown, Dan Klipfer, and I, we in parallel tested these genotypes against the different disease systems. And of course, when we have done that, my test of 2 years, of course, is the longest going. The other ones are either half a year or a year tested, my colleagues do. But when we had all the data together, we compiled that. And out of this selection, and this is very new information still, but we had at least 2 rootstocks here shown on the right and in the center and at the right, that after transplanting and being grafted to Chandler, made after 1 year of growth, very similar growth, like for example, RX1, which as a grafted tree can make pretty sizable trees. So we are in the middle of testing these, and of course, then you say, well, if you have the wonderful stuff, why it's not available yet? Well, because it takes time. We now raise these rootstocks in the nursery. They need to be planted into the nursery, then they need to be grafted. So that alone is already a 3-year process. And then, of course, we have to follow up with testing to see how these perform long term. Now, with that, I wanted to switch gears a little bit into soil treatments and go through several processes that we do there. So one thing that seems kind of logic, but was one observation that I repeatedly make, or one biological principle, is when is our tree the most sensitive to attack by soil-borne critters? Well, that's when it's planted. And in those days that this picture was taken, we still had bare trees, where a lot has gone to potted trees nowadays, of course. But if you look at the left, I mean, that's a tree was just yanked out of the ground. Then we cut it back to the stick, so to speak. We put it into the soil, and now it goes through transplant shock. It tries to make new roots. And if we now immediately have nematodes attacking that, so something chewing on the roots at this early stage of growth, damage potential is horrendous. And many of these damages are really hard to recover from and takes a long time, a lot of resources, and a lot of patience to make something like that recoup. Whereas when you look at the right side, that's a, well, still young tree, but much more mature in that it has now fully anchored in, of course. It has a pretty big root system. And to an extent, it can compensate for something chewing on its root. Of course, there's also limitations for that, because if it's too many nematodes, of course, such tree will also suffer. And so kind of despise this comment, because it's not a very smart way to address it in my mind, but you can all read, but I mean, it sometimes seems to be the attitude, well, I know I have this problem, but I'm just going to ignore it and go right into that. So that's not a very good decision to make, because it is so very difficult to recoup from initial damage. And yes, in many orchards, we do benefit and make them economically feasible or useful to us when we have the early yield. So that's, I guess, one of the big reasons for soil fumigation and for its big importance. So to find new tools, because soil fumigation may not be around for us forever, we went through a screening of chemicals that have some potential. So many times when big companies develop chemicals, they are first developed for the annual world, because there already the treatment of the upper foot of soil can have big impact on improving plant growth. And that way it's never easy, but it's easier than in the perennial world where you want to treat these upper 5 foot of soil. So to test these chemicals, where also the labels are first written for the annual worlds, we use microplots. So we have just culvers of 2 feet diameter, perpendicular, inserted into the ground to 5 feet, and filled with test soils. And then we drench treat them with 6 acre-inches equivalents of water, where this water, every single drop of it has active ingredient of the chemical that we are interested in. So then we can soil sample and check how many of the nematodes survived that. And in this particular case, we planted pre-stratified nemagot seed in there. This was the nursery project where this would be done like this under nursery conditions, and incubated that in the field, looked how many nematodes penetrated the seedlings coming up in the next spring, and then continued checking out the trees. We did that in a whole series of experiments. And yes, you can see on the date, that was already 2015, when we started learning about these things and finding products that were more helpful than others. And so I circled for you here, for example, Salibro, which is a oncoming nematicide, I should say, that in our hands worked well under microplot conditions and also in field trials. We're going to talk about that in just a moment. It's not registered for California use yet, but the registration is coming down the line, I guess. And so it had favorable conditions here, and that's why we continue testing it. Other ones never made it out of the name experimental B or experimental C, and we discontinued testing them because they were just not helpful enough to reduce nematode numbers. One more thing I wanted to point out here is the dominus at 2x. So this was performing very well. Another material that is not registered yet. This is a so-called biofumigant. It's out of AITC, or allyl isocyanate, which is relative of methyl isocyanate, which you all know probably out of Vapem, the active ingredient of it. This is a close relative to that, and it has similar properties as it works in the soil environment. Meaning that it also is not a true fumigant, but needs to be delivered by some other means, and we chose water to do that. Now, many of you by now have heard about ASD, so you know this process that we're just going to briefly discuss here in a moment, but that also worked very well under these conditions that we use to test this material. So since we need to drench apply this, we experimentally need to get it to the site of action. So we put out this system of drip irrigation hose, every foot one line, every foot of the line an emitter, and then we apply our material uniformly to plots. I'm not saying that this is a method that can be used commercially because it's far too expensive, it's far too laborious, logistically challenging. But when we first want to find any efficacy in these chemicals, we need to start where the chemical has the best chance of being successful. So that's why we are using this method. Same thing, same irrigation system is also what we used in anaerobic soil disinfestation. In this process, I was in collaboration with folks from the USDA-ARS, Greg Brown and Dan Klipfel. We spread the substrate, which in many cases was rice bran on the soil surface. We incorporated with a rototiller, installed the irrigation tubing, covered with tarp to exclude the atmospheric oxygen, and then keep it moist for a month before we plant our crops in there. That was very effective, but of course also it's very expensive to do. So this is how the field would look once the treatment is applied. And then routinely, Greg and I and others have used a rototiller to incorporate this material, which of course is good as long as you can put on plastic lines and plastic tarp on top. But those two items add, in my estimate, about $1,500 an acre, which makes it very expensive. So that's why we have for a number of years now experimented. Instead of using plastic as a soil seal, we now moldboard plow the substrate into the ground, which leaves an about 10-inch soil layer that we can compact, and by constantly irrigating with the same amount that we use under plastic, by repeatedly irrigating, can keep it wet and seem to be able to exclude atmospheric oxygen that way. At least our initial data on that are very promising. So we are pretty tickled about that, because although we not always get the same efficacy, and we just now expanded it to heavier ground, we are mostly working in sandy loams here at Kearney, but we are hopeful that we can make it work to reduce the cost effectively with that. Also, to find solutions, while we had put Dominus originally through the strip irrigation system I showed you, we now have adopted a spray blade applicator where you can see these V-shaped sweeps at the bottom, and well, the material supply and the pressurized system that allows us to dispense the material right on the sweep as the sweep is pulled through moist soil at 4 to 5 inch depths, so that it is not immediately gassing out. Of course, you all know the discussions about VOC, the volatile organic compounds. So we have not been able to convince the company or DPR to check this for VOC emissions, but our casual observation is that it shouldn't be a problem ultimately to have too many VOCs coming off of that, because after we apply the material in the horizontal layers throughout the soil environment, we roll the soil and then start our irrigation right away to drive the material down after it has been applied to the soil. Big advantage in my view is, again, these $1,500 an acre that we could save with this. So let's look at some data. So the first that I wanted to show you is from a walnut trial that we did here at Kearney in the sandy loam. And this is the trunk diameter after 2 years of growth in there. And you see, of course, a control in red. You see the more, well, biocidal materials, talon and Dominus in yellow. And then you see a couple of experimental materials in there. So you can see that the Dominus was very favorably at this time, also had the advantage that they got additional post-plant nematicide applications. So that was putting talon EC a little bit at a disadvantage in this particular trial. Yet these differences persevered, and here you see the yield a year later, when all these materials other than the control got a post-plant application in spring of that year. And you can see that Dominus and talon EC were same up as the non-fumigant nematicide, of course, or unfortunately were only same up as a control. So certainly there's some promise in this Dominus if it was adapted and was being accepted for use in California. Here you see from another trial, also in the sandy loam, the nematode numbers after treatment at planting of the new walnut crop. And you see in this comparison that, yes, the control is, of course, the highest. Talon wiped them out. Salibro, the material we talked already about, as a pre-plant drench, also reduced them severely. Velum 1, unfortunately, as such a use was not useful. And then Dominus and the AST with rice bran as treatments with brassica, so where we grew a cover crop over the year, and use that cover crop as a substrate for ASD, also reduced nematode numbers quite effectively. That was unfortunately not fully converted into cyan diameter, so in growth and vigor, where you can see that the Talon C35 treatment by far is the largest, although treatments like with Dominus were coming close to that because of their variability, and also Salibro and Venom 1 were not doing too poorly, but you can see that the one where we used Brassica to produce the biomass for ASD was just not doing the trick. So that would not be a good situation to get into. But it does give us hope so that these different methods are useful for improving management of plant-parasitic nematodes. And I wanted to show you briefly another trial done here at Kearney, where we can see that when you especially look down for Dominus 80, Dominus 80SB and Dominus 40SB, that means either drench applied, the D, and both SB stand for the spray blade. So you can see that this kind of treatment can save potentially some material that we need. So we can cut the rate of Dominus apparently down to 40 gallons to the acre, which is still a lot, granted, but that's where we are right now. And it also shows that drench treatments with non-fumigants like with Salibro have some potential, but it also shows that our improved or modified application pattern for ASD, meaning to plow it into the ground, has potential to reduce nematode numbers. Yet we see that there are residual populations out there. So yes, ASD can reduce pyrrhinitis vulnus in our sandy loams especially. An application is possible. We could save dramatically if we did that. But also for Dominus, there are modifications indicated that we should use. So with that, we think that, yes, we have residual populations. So the call it very effective and slick way, if you will, of using a pre-plant fumigant. Those days may be numbered at some point, and so the tools that we currently develop, and it's to best of my knowledge, the best of them that we can come up with at this time, they may need some additional post-plant application action to reduce nematode numbers. Yeah, so let's talk about post-plant nematicide applications. And so we developed a protocol for that as well, and we did the exact opposite that I'm recommending to you, which is we messed up our orchard entirely, pardon my French, but we did plant into nematode-infested soil, added more nematodes on there, and let the orchard grow for 1 year, which was really hard on the trees because they got very uneven, they were stunted, so we had high nematode pressure encumbering these plants. And in the second growing season, we started applying post-plant nematicides, so that in our preliminary test, we had found that they may be better than others. We applied those and monitored for nematode population densities during the season and measured that plant growth and yield as the trials continued. So these are also long-term studies. And so when we did that, here's just an example of 2 treatments compared to a control, where you go with a pre-treatment where you see in red and green. Sorry if that doesn't show up properly, but you see the Salibro treatment and the Movento treatment that at the beginning of the season, before the treatment was the highest, after 1.5 months, we can clearly see that the Salibro treatments reduced nematode numbers quite a bit compared to the control and also to Movento. And after 3 months, well, the numbers kind of jump around, I guess. But after 6 months, which is then the new dormant season, the numbers are sitting there where you would expect that the numbers are the highest under the control non-treated and are lower under the different chemical treatments. Now, we could not really fully illustrate the huge potential of Movento to be very helpful under these conditions, but these nematode numbers were reduced, yet it was not necessarily converted into yield under Movento treatments. But what was very striking is that when we looked at the root system at conclusion of this trial, this is one from a control. So you see that it's, well, weak tree, considering that this is 4 years old at the time of this excavation. You see that there's hardly any fine roots on there. The anchoring roots are kind of limpy as well. So it's not a very promising tree right here. And then when we looked at a Salibro-treated root system instead, we see, well, much better growth, of course, but we also see that there's a lot more fine roots on here. And so that was mostly visible in the Salibro treatment. It has been visible in several of our trials that always the treatment with this chemical does allow better fine root growth. Now, of course, the question is, how many roots does a tree really need? How many fine roots? There's not a good answer for that, because I have tried to find out in the literature if there's anything said about it, but there isn't. But just for knowing that fine roots are important for the water uptake and nutrient uptake, it certainly will be helpful to improve plant health. So that kind of leads us to the interesting question of how useful is it to use cover crops? So one aim is, of course, you can read the aims here that are under the soil health umbrella. But of course, when you grow a different plant, you are thinking also of managing nematodes. At least I am as a nematologist, because in places of this planet where there's no nematicides available, cover crops is one of the few tools that can be used to reduce nematode numbers. Now, what we need to know when we want to implement them, we need to know about the host status of these cover crop candidates and how soil temperature will impact their development. So as an example, a cover crop that maybe is a decent host for a nematode, but is grown at soil temperatures that the nematode can reproduce, should be more or less a non-problem and be okay to use. Whereas a cover crop that is a pretty good host for one particular nematode, and the nematode can reproduce during the time of year that this crop is grown, could be problem. So the planting window and the use of time of a cover crop in an orchard is extremely important, and certainly needs more research to do it the most effective way. Certainly right now, a lot of it is cover cropping over the winter, and there things are, of course, less favorable for the nematodes. We did find in some work, though, that unfortunately root lesion nematode can continue reproducing under California winter conditions as soil temperatures are concerned. So there are certainly windows for these nematodes where we need to be vigilant about what crop we use. Then, of course, management strategies is, are we mowing, are we technically, chemically reducing cover crops, are questions that are out there. And so from the nematode standpoint of view, we did a screen of different brassica species for the susceptibility to Patholenchus volus. And if you look here, most of the crops are, well, much lower than Nemagard, our preached rootstock standard in here that is highly susceptible to root lesion nematode, but they do allow for some reproduction on there. And so some of them allow a lot less. So those are certainly some that are better to take for such a strategy to try to manage nematodes in the field. The picture is different when it goes against root knot nematode, where the variability is much higher. And although the nematode numbers are not extremely high that developed in our greenhouse assay on there, you can see that some of them reproduce quite a few egg masses on there, so new nematodes, if you will. And so when we choose a cover crop, we need to consider that in our choices. Because if we grow a cover crop at times that this nematode could reproduce, we should stay away from cover crop species that would allow its reproduction. And so one first strategy that we used with cover crops is that we came right after almond harvest, mid-September maybe, that we planted our cover crops into the orchard. And we initially did that by leaving also a spray strip around the tree, not as much as a grower would like it, but we wanted the roots of the cover crops to be at least in the vicinity of the roots of our tree out there. And so it is possible. And yes, if we do this in September, of course, you need additional irrigation, and later in the year, you may be fine. Now, the beauty of that was that when we cropped our cover crops like this in the almond orchard, we did get a reduction of nematode numbers. You see in red the control at 0 to 3 feet depth. Then you see a Salibro treatment at 0 to 3 feet depth. And then you see Merced rye that was more or less grown in the middles, but yet we saw benefits in some reduction of nematode numbers. The same token, we also saw that the water infiltration rates by Merced were much higher, meaning it took less time for the water to percolate the soil. That may sound trivial, but in times of drought, everybody worries about not having enough water to grow anything. But if you could capture the winter rain much better, meaning the soil takes the water much quicker, that will be helpful. Also, if your irrigation water goes into the soil much quicker during summer, then a lot of it evaporating before it can ever get into the soil, that will be helpful as well. So I think there's a lot still to be learned about cover crops. So we carried that further and saying that, well, maybe we can grow nematode antagonistic plants so close to the root zone of almonds that we reduce the negative effect of nematode infections. And to do that, we installed a pretty sizable trial where we planted our cover crop right next to the tree row, as shown here in the upper left-hand side, and found that a cover crop used that way of radish was not really helpful to support the growth of almonds. As a matter of fact, the trees are looking really poorly and do that throughout the trial. Because after fumigation, shown on the right-hand corner, trees were growing much better, whereas the radish more or less eliminated the almond crop, one would always have to say. So now this is not to dash radish altogether, because it may be in the conditions that it's typically grown, meaning in the middles, it has its place for improving water infiltration rates and others. But as the nematode management tools under our hypothesis, it wasn't really all that helpful. You can see here rye, or another crop is Codalaria, you see on the right-hand side. So there are different cover crops which are milder, let's say, on the main crop almond. And so important is when you make your own experiments, don't assume because it looks good in one area or because the neighbor does it, it will work for you. Radish was negative for plant growth. So, and when used in the way that I just showed you, grown very close to almond trees, whereas other cover crops have maybe more potential. And we are just at the beginning of all these studies, so we don't have final data on that yet. And so far, we were not able to outperform the suppression by chemical amendments rather than with our cover crops. So still a ways to go. So I hope today I showed you how plant-parasitic nematodes cause damage. We will all remember that there are not only bad nematodes out there. There are many that are very important for us. The free-living nematodes that are important in nutrient breakdown and the nutrient cycling in the soil. We learned how to monitor for plant-parasitic nematodes. That yes, it's best to have a live plant there so that we can sample out of the root zone of this living tree that we are interested in. Even though we may want to pull this orchard out, it's still a good time to sample before we do that so that we have the best site to have a chance to detect the nematodes. We learned that if this field has been fallow for too long, nematodes can possibly survive in greater soil layers, and the fallow alone will not reduce nematode numbers sufficiently that it's really sufficient to protect our new planting. Rootstocks can mitigate nematode problems, and yes, there's constant work ongoing on that. Unfortunately, it's a very long-going process before new rootstocks come online, because these need to be tested thoroughly. They need to be tested for the nematode status, but of course also for the horticultural characteristics. We learned about some soil treatments coming online. We are not quite there yet, I guess, to have them fully available, but we are certainly hopeful. We talked about some post-plant mitigation options, and we briefly visited the topic of cover cropping or companion cropping in an orchard. All this work is not done by myself, of course. It's always not only by myself, but in team efforts, as shown here, a whole bunch of folks, and I'm sure I forgot at least half of them who should have been up there. So it's collaborative work with different aspects of tree health. I'm very happy that I have a strong nematology research team here. at Kearney, but also that I can interact with my colleagues at Riverside, different collaborators, of course, the farmer collaborators, UC and our collaborators. It's extremely important for my work. The tree nurseries who help me out, Trical who helps me out with some trials, and none of this would be possible without the generous support of the different entities that you see down here. So with that, we try to move forward and find solutions so that you can continue to have productive orchards in the future, although nematodes will not go away anytime soon. So with that, thank you very much. And I think we have time for some questions, right?
How is the plant competition separated out from nematode suppression when using cover crops?
Yeah, that's an excellent question. So we try to compensate as much as we can by at least controlling the water. So every one of our cover crops has cover crop plots has separate irrigation. So we monitor the water status of the plant by stem water potential to make up for the different water amounts that the cover crop may extract that is not available to the tree. But they are on the same water status, so it's not only competition in that sense. There may be other things ongoing, which we are exploring. And so I do not want people to go away of thinking that the radish increased nematode numbers, and that's because the tree doesn't grow. No, it's because of this mentioned plant competition.
Okay, and I'll just ask just one last one here. Are cover crops better for a healthy soil in the long term versus chemical treatments?
It certainly would appear so. I mean, a healthy soil, independent of what name that name has been given to those kind of soils now, it will need some balance of organic matter for water holding capacity and for water infiltration. And so this additional rooting to the soil will make the soil ideally something closer to, call it a sponge, where water can flow in easier, where air can also get in for root growth. So ultimately, it truly will be a healthier soil to have more cover crops in there. We are living in a desert climate where organic matter is typically burned up fairly quickly. So if we don't constantly feed the soil this, it will be difficult to maintain a really, truly healthy soil.
All right, so I'm going to go ahead and close out. Thank you again for your presentation. Appreciate you staying on. And thank you everybody for attending today. And we hope we will see you all at the next one in 2023.
Well, thank you.